Welcome everybody to the second webinar of our T-cell series. T-cell culture, fine-tuning your T-cell research workflow step by step. In the next 35 minutes, you will learn all about the best T-cell culture products by Miltenyi Biotec. So enjoy. Please acknowledge the displayed disclaimer. So let's begin with a brief company presentation. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi himself and is the largest owner-managed company in Germany. We have two main sites in Germany, one in Bergisch Gladbach, where the headquarters are located, and one in Teterow, where the GMP manufacturing facility is located. We have subsidiaries in 28 countries and distribute to additional 73 countries. Currently, we employ over 3,000 employees worldwide. Our mission is to serve the needs in life science research and to translate new scientific findings into clinical applications. As for the agenda, first we will have a brief look at the biology and function of T cells, as well as at the concept of workflows and the hallmarks of MACS cell culture. Next, we will have a look at our highlight T cell culture products. We start with media and cytokines for T cell expansion and polarization. Then we will learn about T-cell stimulation reagents for polyclonal, but also antigen-specific T-cell stimulation. And finally, we will talk about MACS GMP quality grade as ideal tool for T-cell manufacturing in a clinical context. Let's start out with a brief introduction about the biology and function of T-cells. A quick look at T-cell biology covers the basic aspects. When talking about T-cell phenotypes and function, mostly surface markers, cytokines, and transcription factors come into play as defining molecules. T-cells hereby play as effectors of the immune system an integral role in the adaptive immunity. They can be subdivided broadly into several phenotypically and functionally distinct subsets such as T helper cells, cytotoxic T killer cells, T regulatory cells, as well as the rather heterogenic group of unconventional T cells, comprising, for example, natural killer T cells and gamma delta T cells. T cells are the only immune cells that are able to form a long-lasting immunological memory that helps to respond faster and more efficient to already experienced threats. Key surface markers that are always mentioned in regards to T cells are the T cell receptor TCR that recognizes antigens, as well as the activating co-receptors CD3 and CD28. Those three surface markers are present on all T cells. Furthermore, co-receptors CD4 and CD8 are the defining markers for T helper cells and cytotoxic T killer cells, respectively. To exert their function as effectors of the immune system, T cells release cytokines to communicate with their environment and to cause various effects, such as promoting inflammation. These cytokines include interleukin 2, interleukin 4, interferon gamma, tumor necrosis factor alpha, transforming growth factor beta, and interleukin 10. T cells are most relevant in both maintenance of health as well as fighting diseases. Last but not least, these properties make T cells ideal tools for immunotherapy, where T cells, for example, are genetically modified as CAR T cells to attack cancer specifically. Although our best-known products are indeed the microbeads and isolation kits, we are not just a cell separation company. We actually cover the whole experimental T-cell workflow from cell separation to cell analysis, including also sample preparation as well as cell culture. We provide thereby solutions for each step of your experiment. And today we concentrate on T-cell culture. Well, although it may be now easier to get an overlook of our product world, we thought maybe the best way to arrange our large T-cell portfolio are distinct workflows specifically designed for your experimental needs. Thereby, we cover as focus workflows, amongst others, regulatory T cells, CAR T cell research, gamma delta T cells, tumor-infiltrating leukocytes, polarization of T cells, antigen and virus-specific T cells, and of course activation and expansion of T cells. With these comprehensive workflows, you can easily choose the products relevant for your specific field of interest. Please keep in mind the T-cell portfolio is growing by the day and the respective workflow overviews are all work in progress, so check regularly for updates. By the way, you may notice throughout the presentation small yellow buttons paired with a little click hand symbol, just like the one on the lower right. In the PDF version of this talk, you can access the respective product pages, marketing materials, videos, and protocols by simply clicking these buttons, as they are all hyperlinked. Getting your whole workflow from Miltenyi Biotec means ease of mind. Standardization, reproducibility, and organization of an experimental workflow become much more feasible. Miltenyi Biotec, as supplier for the whole T-cell workflow, means that all reagents fulfill the same high-quality criteria and the order process can be streamlined. Thereby, harmonizing the steps of an experiment as well as automation and upscaling are easily performed. Getting all products from Miltenyi Biotec guarantees smooth procedures and less errors, since, for example, our cell separation is tested for compatibility with our flow cytometry and our media for compatibility with our cytokines, and so forth. Moreover, having Miltenyi Biotec as full supplier for the whole workflow means also getting full support for every step of your experiments thanks to our great scientific support. Moreover, a hallmark feature of Miltenyi Biotec's workflow solutions is the seamless translation from a research setting into a clinical setting and back, thanks to our MACS GMP portfolio, as well as cell manufacturing platforms such as the CliniMACS Prodigy. Now let's see what makes MACS cell culture products so special. Cultivate your vision. When culturing T cells, apart from activation and expansion, also differentiation into different T cell subtypes is an important topic. The latter is performed with cytokines and functional grade antibodies. Activation is performed with either polyclonal stimulation reagents that, for example, engage CD3 and CD28, or antigen specifically, using, for example, our Pep Tivator Peptide Pools. Expansion culturing is best performed in special T-cell media, such as our serum-free TexMACS medium. In the following slides, we will have a more detailed look at Miltenyi Biotec's T-cell culture portfolio. Of course, for cell culture, quality is the main criterion. However, what does that mean? Let's look at it from the angle of the challenges a researcher has to face when performing T-cell culture. Reproducibility is essential to successfully generate results and to publish. Time pressure is thereby a big issue, and repeating failed or non-reproducible experiments has to be avoided. Indeed, time is money, so time saving goes along with cost saving, another big challenge. But saving costs means also correct dosing of reagents without wasting, especially when working, for example, with delicate primary cells. That is why we provide MACS cytokines with a lot-specific activity that allows exact dosage. Cell culture reagents, furthermore, need to meet certain regulatory requirements to be translatable into a clinical setting. With all that in mind, the MACS cell culture portfolio indeed provides best quality. But how do we provide the best quality with MACS cell culture reagents? First, Miltenyi Biotec cell culture reagents are optimized to support complete experimental workflows without gaps to allow for consistently high quality throughout the whole experiment. Second, we have a strong focus on standardization since reproducible and reliable results are the key for success. And third, we provide reliable tools for the development of cellular therapies with our MACS GMP product line. Let's now have a look at our highlight T-cell products, starting with media and cytokines. Of course, culture media play for the cultivation of T-cells a pivotal role. Here we offer our serum-free TexMACS T-cell medium. Free of animal-derived components, it is available from research grade up to MACS GMP grade and ensures optimal T-cell growth, activation and expansion. Tedious pre-testing of expensive and rare fetal bovine serum is not needed, and undesired side effects caused by the serum are eliminated. Originally, TexMACS was developed for the clinical-grade, serum-free manufacturing of human CAR T cells. However, TexMACS in research grade is of course also beneficial for many other T cell applications, especially the human ones. These include polyclonal T cell expansion, expansion of regulatory T cells and gamma delta T cells, T helper cell polarization, and CAR T cell manufacturing. Moreover, also for the polarization of mouse T cells, TexMACS has been proven to yield superior results when compared to the standard basal medium. The serum-free TexMACS T cell medium allows for most consistent T cell culture results. With the MACS GMP version and of course our expertise, translation into clinics has never been easier. For best activation and expansion, our specific T cell medium TexMACS and our efficient activation solutions, the T cell activation and expansion kit and T cell TransAct go hand in hand. As you can see on the left graph, TexMACS outperforms serum-free competitors and RPMI alike in a 14-day human T cell culture after activation, with the T cell activation and expansion kits reaching over 80-fold expansion without the addition of serum. On the right side you see how efficiently TexMACS and TransAct combined work in a completely serum-free, fully translational cell culture setting. After 14 days of culture, human T cells activated with TransAct and serum-free TexMACS even outperform a competitor's classical bead-mediated T cell activation in RPMI with 10% fetal bovine serum, being thereby more reproducible, convenient, and of course translational. A highlight application is hereby the activation and expansion of human gamma delta T cells with our TexMACS medium as well as our cytokines. As you can see, human gamma delta T cells nicely expand and are kept pure over the course of 15 days when using TexMACS GMP medium supplemented with 100 international units per milliliter of our MACS GMP grade interleukin 2 and interleukin 15 as well as 5 micromolar of solendronate. To point out again, with TexMACS different quality grades, such as research grade and MACS GMP grade, everything from basic research in academia to commercial manufacturing in pharma industry is covered. TexMACS media are high quality products manufactured with superior quality standards and extensively tested, serum and xeno-free. Ideal for a seamless translation from basic research to clinical scale self-manufacturing. When talking about T cell culture and media, naturally, also cytokines and functional grade antibodies have to be included. Miltenyi Biotec offers a huge variety of cytokines and functional grade antibodies to allow for any kind of T cell culture conditions, from stimulation to expansion to polarization. This comprehensive overview, for example, shows the different T helper cell subtypes and how they can be polarized in vitro using our MACS cytokines and functional grade antibodies. It also shows how the respective phenotypes can be assessed using our MACS flow cytometry portfolio. Consequently, I'd like to point out that apart from our polarizing cytokines and functional grade antibodies, we also offer flow antibodies for the according analysis of the different T cell phenotypes via surface markers, cytokines, and transcription factors. Thereby, our cytokines and analysis tools are not only limited to T cells, but we offer much more varieties, such as for dendritic cells and NK cells as indicated on our webpage. Please note that we offer many stimulation reagents and cytokines in different quality grades, namely research grade, premium grade, and MACS GMP grade according to your needs. Let's look deeper into our premium grade cytokines. For T cell culture and cell culture in general, it is important to use the optimal amount of cytokines. Several publications have shown that cytokine concentrations have of course an impact on cell phenotype and function, so proper cytokine dosing is essential for reliable results. Cytokine levels in cell culture assays should be saturated, as shown in the curve on the lower left. Yellow and red arrows indicate insufficient cytokine concentrations. If the assay is not saturated, this may cause fluctuations in cellular responses. The green arrows, on the other hand, indicate saturated cytokine levels to achieve optimal cellular responses. Interestingly, in the saturated area, 100 international units per milliliter and 1000 international units per milliliter work equally well. In consequence, failure to add the correct amount of cytokines, be it not enough or too much, will either lead to suboptimal results or a waste of reagents. Moreover, cytokines from different production lots that are dosed only based on protein weight, for example nanograms per milliliter, result in a high variability in cellular responses. As the graph right next to the curve shows, results are reproducible when cytokines are dosed based on activity units, for example international units per milliliter. So, how can you achieve reliable and constant cytokine levels for your experiment? The most important factor for correct cytokine dosing is the specific cytokine activity. Cytokine activities are measured using a bioassay to determine how potent the cytokine is in inducing a cellular response. This specific activity can vary between production lots because the amount of properly folded and active cytokines is not constant. For our premium-grade cytokines, we test the cytokine activity for each production batch and provide the results in a certificate of analysis. With these values at hand, you can precisely dose your cytokines based on activity and achieve reproducible cell culture conditions. In addition, at Miltenyi, bioassays are calibrated with international reference substances whenever such a standard is available. This means that the reference substance with known activity is run in the same assay as our product. This allows to obtain reliable calibrated cytokine activities. As you can see in the graph in the middle, all this results in consistently higher cytokine activity when compared to non-calibrated competitors. For those working with clinical research, the lot-specific activity is of course also provided for all MACS GMP cytokines. MACS GMP cytokines additionally feature extensive documentation, lowest endotoxin content, and highest purity, ideal for relevant preclinical studies and easy translation into clinics. Here is an exemplary overview of our highlight human premium-grade T-cell cytokine products. However, of course, also many mouse versions are available. All relevant cytokines for T-cell expansion and polarization are available in research grade, premium grade, and even MACS GMP grade. Also, beyond cytokines, there is a multitude of T-cell activation and stimulation reagents that you can find on our website and that are worth looking into. Talking about convenience and consistency, the CytoBoxes mouse are kits that allow for the easiest polarization of naive mouse CD4 positive T cells into Th1, Th2 and Th17 subtypes without complicated ordering and calculation of single polarization cocktail components. The CytoBoxes contain in one convenient kit everything needed for the efficient mouse T helper cell polarization, including cytokines and functional grade antibodies. As you can clearly see in the presented data, Th1, Th2 and Th17 cells are successfully polarized and express their respective signature cytokines and transcription factors such as interferon gamma, interleukin 4, IL-17, as well as Tbet, GATA3 and DOA gamma T respectively. As you can see below, best results are of course generated with our special T-cell medium TexMACS. Next, let's have a look at our stimulation reagents in particular. We discriminate between two different T-cell stimulation approaches. Polyclonal stimulation that activates T-cells regardless of specificity, and antigen-specific stimulation that activates only T-cells of a certain specificity. The first one is achieved usually via antibody-mediated CD3-CD28 engagement or via a so-called superantigen. The latter one is achieved via certain antigens such as peptide pools. Let's start with conventional polyclonal T-cell activation and expansion. Our classical bead-based expansion reagents, the T-cell activation and expansion kits, offer hereby reliable yet flexible polyclonal T-cell activation. Of course, specific T-cell activation and expansion kits for mouse and human T-cells and Tregs are available. The employed MACS beads can thereby be flexibly loaded with biotinylated antibodies of your choice to optimize activation stimuli. As seen in the graphs, both mouse and human expansion rates surpass very well 100-fold for both CD4 and CD8 positive T cells. T-cell activation expansion kits are not only consistent in their performance, but also very economically priced, especially when considering their high capacity per kit. Please let me also point out that we have a non-human primate version available. Let's have a closer look at the T regulatory cell variant of our expansion kits, one of our highlight products. They are available as human and mouse versions. Apart from the high expansion rates, in this example up to 40-fold within 21 days, I want to point out that the Treg expansion kits guarantee a stable FoxP3 expression even during prolonged expansion periods. It is a well-known problem that Tregs lose FoxP3 expression in culture. As you can see in these dot plots, even after 20 days of expansion, the frequency of FoxP3 positive Tregs is very similar to the starting frequency. Again, please find the corresponding application notes and protocols on our website. When talking about Treg culture, also Treg suppression assays are an important topic to address. With our Treg Suppression Inspector Human, we offer an optimized all-in-one solution to measure human Treg suppressive capacities. The kit offers maximum convenience and easy handling with striking results, again perfectly harmonizing with the TexMACS medium. Just co-culture Tregs and T responder cells activated with the activation reagent provided by the kit and assess how your Tregs suppress T responder cell proliferation in different ratios. Also here, please find the corresponding application note on our website in the T-cell application section. Our next polyclonal T-cell activation and expansion reagent is the absolutely innovative T-cell TransAct Human. It is a polymeric nanomatrix that efficiently and reliably activates T cells via CD3 and CD28 engagement, as you can see in the dot plots on the right, displaying strong upregulation of activation markers CD25 and CD69 in both CD4 and CD8 positive T cells after stimulation with TransAct. As you saw some slides before, expansion rates hereby exceeded 100-folds in the course of 15 days. As a nanomatrix, TransAct is as convenient as it gets, ready to use, easy volumetric dosage and simple removal by washing. So no tedious debeading step is necessary, which makes TransAct compatible to automated cell manufacturing systems and minimizes cell loss. Being available also in MACS GMP grade, TransAct is the ideal T-cell activation reagent for automated and streamlined translational applications such as CAR T-cell manufacturing on the Clinimax Prodigy. So, TransAct resolves quite some pain points right away, easy to handle, reliable and consistent in performance, as well as easy to translate into clinics. So, we learned about two different polyclonal activation reagents: the bead-based T-cell activation and expansion kits and the nanomatrix-based T-cell TransAct. Both efficiently activate and expand T cells, but which one to choose when? The T-cell activation and expansion kits allow for maximum flexibility with the option to load individually antibodies of your choice. Furthermore, distinct mouse, human and non-human primate T-cell and Treg versions are available. This flexibility is especially interesting for basic researchers that need a broader application spectrum. T-cell TransAct, on the other hand, is extremely convenient to use and easy to translate into clinics, but is restricted to the activation of human T cells via CD3 and CD28 engagement. This makes TransAct especially interesting for translational researchers, for example in the CAR T-cell field, but also to basic researchers that need less flexibility but higher convenience. Our third and last option for polyclonal T-cell stimulation is CytoStim, the non-toxic Staphylococcus enterotoxin B, in short SEB, alternative. CytoStim cross-links the T cells TCR to the antigen-presenting cells MHC. This means, as with SEB, the T cells need the presence of antigen-presenting cells to be stimulated properly with cytostim. This is best achieved in a PBMC setting and may serve, for example, as a functional control in antigen-specific T cell research. Since the receptor cross-linking by cytostim is antibody-mediated, no toxic superantigen like SEB is involved. However, the efficiency is very well comparable to SEB, as you can see in the dot plots below. So with cytostim, no complicated to handle and dangerous reagent is needed, but we offer a simple to use and efficient alternative. As pointed out before, another way to activate T cells is the antigen-specific stimulation, for example using peptide pools. Please remember, this works best in the presence of antigen-presenting cells, for example in a PBMC context, or in a co-culture of purified T cells and antigen-presenting cells. Hereby, the most efficient antigen-specific stimulation of T cells is achieved reliably with our Pep Tivator Peptide Pools. These extensive panels of virus, tumor, fungi, microbiota or autoantigens consist of 15 mere peptides with an 11 amino acid overlap, covering hereby the complete sequence of the respective antigen. The peptide composition ensures the optimal stimulation of both CD4 positive and CD8 positive T cells. Pep Tivators are available in different quality grades as well as in a convenient 96-well format for high throughput applications. The different quality grades include research grade, premium grade, and MACS GMP grade. Most notably, we also added SARS-CoV-2 Pep Tivators to our portfolio to support your virus-specific research best. Please follow the respective links to get to the Pep Tivator product pages as well as to see an informative SARS-CoV-2 Pep Tivator video. Please note that we also offer TLR ligands as well as recombinant proteins for cell stimulation. In this nice example, we stimulated PBMCs of SARS-CoV-2 reactive donors with the aforementioned SARS-CoV-2 Pep Tivator peptide pools as indicated. A mix of CMV EBV Pep Tivators was used as positive control. We pre-gated then either on CD4 or CD8 positive T cells and assessed CD154 and TNF-alpha expression as well as CD69 and interferon gamma expression respectively on the MACS Quant Analyzer 16. As you can see, the reactive donors T cells respond specifically to the stimulation in comparison to the negative control with the expression of the respective activation markers and cytokines. Last but not least, an outstanding cell culture reagent in our portfolio is the transduction enhancer Vectorfusin-1. After activation, you might want to transduce your T cells, depending on your workflow, for example in the context of CAR T cell research. Vectorfusin-1 is hereby particularly handy. This non-toxic histidine-rich amphiphilic peptide enhances efficiency for lentiviral and retroviral transduction and is compatible with static transduction and with spin oculation. Conveniently, no pre-coating is needed. Vectorfusin-1 is simply added to the T cells and, as seen here on the right, thereby boosts drastically transduction rates. With being available up to MACS GMP grade, Vectorfusin-1 is ideally suited for translational applications such as CAR T cell manufacturing. Finally, let's discuss our outstanding MACS GMP grade cell culture reagents. MACS GMP quality provides the respective products with all needed certificates and documentation, as well as supplement information and descriptions to facilitate ex vivo processing for cell manufacturing. MACS GMP is thereby backed up by a certified quality management system to ensure highest quality standards. MACS GMP comprises T-cell activation and expansion reagents, cell culture reagents such as cytokines and media, as well as culture and cryopreservation bags as part of an end-to-end approach to cell therapy research with proven performance throughout the complete manufacturing process. Talking about quality, all MACS GMP media are manufactured and tested to the highest quality standards for optimal performance, consistency, and safety. This means that they are animal component free, produced in a strictly controlled environment and undergo extensive QC testing with provided documentation for traceability and qualification of raw materials. As an added bonus, I'd like to point out some highlighted T-cell applications that are covered by our full workflows, of course including MACS cell culture. So, in the following, I will present very briefly 5 workflows that specifically benefit from the superior quality of MACS T-cell culture reagents. Of course, we have more than those 5 T-cell workflows on our webpage, so please visit the T-cell application pages via the links below. So let's start with our T-cell activation and expansion workflow, which is of a pivotal importance for so many T-cell applications. Thereby, reliable and efficient cell culture results are essential for the success of the intended downstream application. In our respective activation and expansion workflow, first, T-cells of a given subtypes are isolated with highest purity using our MACS microbeads and isolation kits. Then, the T-cells are activated and expanded, for example with the T-cell activation expansion kits or TexMACST, TexMACS medium, as well as MACS cytokines such as IL-2 premium grade. Lastly, the phenotype and activation status is analyzed using, for example, reaffinity antibodies and the MACS flow cytometer. Next, let's have a look at the CAR T-cell research workflow, which is of course fully translatable into clinics. In the CAR T-cell research workflow, first, CD4 positive and CD8 positive T-cells are isolated using our MACS microbeads. Then, the T-cells have to be activated before transduction using TransAct and expanded in TexMACS in the presence of MACS cytokines such as IL-2 or the combination of IL-7 and IL-15. Hereby, not only highest performance is essential, but also reliability, reproducibility, and the easy translation from a research setting into a clinical setting, which is typical for CAR T-cell research. In the end, but also already during ongoing manufacturing, the phenotype and functionality of the resulting CAR T-cells is assessed using our flow portfolio, for example with reaffinity antibodies, the MACS cytokine kits, and of course the MACS flow cytometers. Another very well culture-driven workflow is the polarization of CD4 positive T helper cells. Naive CD4 positive T helper cells are gently isolated using our naive T-cell isolation kit. For the polarization into the various T helper cell subtypes, of course our MACS cytokines and TexMACS medium are the best choice. And then again, the phenotype and cytokine secretion can be assessed using our reaffinity antibodies, the MACS cytokine kits, and the MACS flow cytometers. We also offer a complete portfolio and workflow for T regulatory cells. We have several Treg isolation kits to isolate highly pure human and mouse T regulatory cells. Tregs are then cultured in TexMACS and expanded, for example, with our Treg expansion kit. The phenotype and suppressive capacities can then be assessed using our reaffinity antibodies and the Treg Suppression Inspector on the MACS flow cytometer. And lastly, we also offer an optimized workflow for rare antigen-specific T cells. First, isolate highly pure PBMCs without density gradient centrifugation. Next, stimulate the T cells within the PBMC context antigen specifically, in this case with our SARS-CoV-2 Pep Tivator Peptide Pools. Finally, analyze the phenotype of the antigen-reactive T cells using our flow kits and the MACS flow cytometer. Although this was quite an extensive overview of the T-cell culture portfolio, you have to remember just 3 easy things. First, we offer more than just T-cell separation. Second, we cover the whole T-cell workflow. And third, we offer a solution for every pain point. So have all the success with our innovative and complete T-cell portfolio, as displayed here in this fully featured overview. Again, please keep in mind the T-cell portfolio is growing by the day, and the respective workflow overviews are all work in progress, so check regularly for updates. Please follow also the links on the lower right to access the respective T-cell application and product pages, as well as to download our T-cell brochures. Talking about more information. Of course, you can find a variety of helpful marketing material downloadable from our webpage www.miltenyibiotec.com. For T-cell specific applications, just click on the button on the lower right side. This includes flyers and brochures, webinars and videos, protocol and data sheets, customer reports, application notes, scientific posters, and datasets. And of course, you can always directly reach out to the responsible sales representative in your area. I hereby end this presentation with this nice picture of our company campus in Bergisch Gladbach at night. So thank you very much for your attention. And please don't forget to visit our website www.miltenyibiotec.com.